Alzheimer's disease is a growing global concern, especially as populations age. Scientists have identified the APOE4 gene as a major risk factor for late-onset Alzheimer's, while APOE3 is considered neutral. But genetics isn't the whole story. Lifestyle, particularly diet, plays a crucial role in shaping health outcomes. Recent research using humanized APOE3 and APOE4 knock-in mice explores how a high-fat Western diet impacts both metabolism and retinal health. By comparing mice fed either a standard control diet or a Western-style high-fat diet over several months, researchers can tease apart the complex interactions between genes and environment. This approach helps us understand why some individuals are more vulnerable to metabolic and neurodegenerative diseases, and why personalized dietary strategies may be necessary for those at higher genetic risk. Study found that a Western diet quickly triggered metabolic changes in APOE4 mice. After just 2 months, these mice developed glucose intolerance, a precursor to diabetes, while APOE3 mice only showed similar issues after longer exposure. Interestingly, APOE3 mice on the Western diet developed more severe hyperlipidemia, elevated blood lipids, than their APOE4 counterparts. Suggests that while APOE4 mice are more sensitive to early metabolic stress, APOE3 mice may accumulate more systemic lipid problems over time. These findings highlight that the same diet can have different effects depending on genetic background. The research underscores the importance of considering both genotype and diet when assessing metabolic disease risk, and suggests that interventions may need to be tailored accordingly. Beyond metabolism, the Western diet also affected the eyes, specifically the retina. APOE4 mice on a control diet already showed early retinal thinning, but those on the Western diet first experienced retinal swelling, then degeneration after prolonged exposure. Vascular analysis revealed that APOE4 mice developed increased vessel tortuosity and reduced vein width, especially under the Western diet. These changes occurred without visible retinal lesions, indicating that dysfunction happens at a structural and functional level. Visual performance tests showed that APOE4 mice had progressive declines in visual acuity and contrast sensitivity, especially when fed the Western diet. These findings suggest that APOE4 carriers may be especially vulnerable to diet-induced retinal and vascular impairments, reinforcing the need for targeted prevention strategy. Key discovery was the role of inflammation in diet-induced retinal dysfunction. APOE4 mice fed a Western diet showed elevated levels of IL-1B, a pro-inflammatory cytokine linked to both Alzheimer's and diabetes. This inflammation was closely associated with retinal and vascular impairments, even when other inflammatory markers remained unchanged. Functional tests such as electroretinography revealed that APOE4 mice had reduced retinal responses and bipolar cell deficits, especially under the Western diet. These results point to a vicious cycle where genetic risk, poor diet, and inflammation amplify each other, accelerating neurodegeneration. The study's findings emphasize the importance of genotype-specific dietary strategies to reduce the risk of metabolic and neurodegenerative diseases, especially for those carrying the APOE4 allele. Summary: this research demonstrates that the impact of a Western diet on metabolism and retinal health is strongly influenced by genetic background. APOE4 mice are particularly susceptible to early metabolic and retinal dysfunction when exposed to high-fat diets, while APOE3 mice develop more pronounced lipid abnormalities over time. Importantly, even a control diet does not fully protect against retinal changes in genetically at-risk mice. These findings highlight the need for personalized nutrition, diets tailored to an individual's genetic makeup, to effectively prevent or mitigate the risks of metabolic and neurodegenerative diseases. As our understanding of gene-diet interactions deepens, targeted dietary strategies could become a powerful tool in promoting lifelong brain and eye health.